Hi. Hydrolytic damage is continuous. Water is everywhere. There are reactions, and that means the genetic information is also getting affected, like deamination or depurination. So there must be active mechanisms from the side of the cell to keep or repair these damages. And that is what we will discuss today. I am Dr. BCMR. So deamination of cytosine could result in the formation of uracil within DNA, which is abnormal. Similarly, deamination of methyl cytosine could result in thymine, which is normal, but which would establish as a mutation. So there should be active mechanisms of how these the uracils, which are deaminated cytosines, should be removed. Not just uracils. There are many other bases that should be removed. Any kind of changes, abnormalities, or even addition of some groups, they should be deleted. And the one prominent one is, or repair mechanism, is called as base excision repair. As the name suggests, the base excision, which means that we are going to, or the repair mechanism happens to work by removing the whichever base is abnormal or whichever base should have been removed. So first thing, we will take a DNA. Here is the DNA with phosphate ribose 5' to 3'. That's the backbone. And here we have bases. Assume that this is the cytosine, which is deaminated cytosine. So now it's uracil, and that's not normal in DNA and should be removed. So there are glycosylases, which are enzymes that break the glycosidic bond. That's why we call them as glycosylases. And here the enzyme that is working is uracil glycosylase, because it is specific to identifying uracil within DNA and breaking the glycosidic bond there. So we also have other types of glycosylases. It is dependent on the kind of abnormal bases, nitrogenous bases, that they can detect and remove. So it works very simple. It first breaks off the glycosidic bond, thereby releasing the uracil. So the enzymes have specificities for that particular base only, and it will be amazing to understand or appreciate that the difference between thymine and uracil is just one methyl group, and these specific glycosylases, that is here it's uracil glycosylase, will only bind to uracil but not to thymine. And then we also have, once that is removed, we have a site here which is kind of abasic site, and that means there is no base here, and that is an abnormality as well. Once the glycosylase releases the uracil and moves away, then there is another enzyme called as AP, endonuclease exonuclease. You can call it as apyramid endonuclease exonuclease or apyramidic. It doesn't matter, but AP endonuclease. So it has two activities. First one is it has an endonuclease activity. Endonuclease activity is about cleaving within the DNA. Exonuclease, of course, is cleaving from the ends of it. So by the first endonuclease activity, it cleaves in the middle of the DNA where the abasic site is present, and then using exonuclease activity, it will remove one additional base. So there will be a cleavage site here, and there will be a cleavage site here. Thereby, this whole nucleotide would be removed, and as a result, we have a broken DNA, something like this, with one base. Now, if this is the 3' end, DNA polymerase 1 could bind here, and it will start polymerizing until the gaps are filled, and at one point the ligase can seal the backbone. That is how base excision repair works. Simple, remember. The important enzymes are glycosylases. Each of the bases, modified or abnormal bases within the DNA that frequently form are identified by their respective glycosylases, breakage of glycosidic bond and release of the base, and then formation of a basic site. You can call it apurinic or apurinic site, and then endonuclease and exonuclease will cleave that one base with the ribosome. The deoxyribose phosphate will be removed. There is a space for one nucleotide which can be filled up by DNA polymerase and sealed by DNA ligase. It's a simple one. Thank you very much.